All yours. All right, so where's the presentation?
Sound okay,
Okay,
screen
everyone.
okay.
Good to know. Okay, so the project's cognitive storm, long as Newton's stoichiometry transformation for optimized resource management. So why is this important? So how the agricultural production can improve sustainability? We know already that we have to be less dependent on synthetic fertilizers in the long run. We actually have to reduce our fertilizer application as the European targets 2030 by 20%. We have to improve nutrient use efficiency. The EU target is improving by 50%. So we want to minimize soil erosion. We want to reduce the loss of soil fertility and biodiversity. We want to make sure that we have fewer groundwater pollution and greenhouse gas emissions. So all the political aims are going in that direction. In order to achieve that, we have to look at different approaches. So we also have here one approach, where we want to protect agricultural production and yields with reduced dependence on synthetic fertilizers, and stay within the limits of the current and future framework directives, including nitrates directives. And so that one of the things we want to look into is wider adoption of the circular economy, using fertilizers cycling derived from these products, as well as promotion of soil microbiota functionality to make nutrients plant availability in order to increase the nutrient use efficiency. So the objective of Storm is to improve the sustainable agriculture via optimizing soil carbon stocks. So we not only want to improve soil carbon stocks in order to lock carbon into the soil from the greenhouse gas emissions point, but in this case also to improve fertility and agriculture. We want to reduce dependencies on the linear fertilizer economies. We want to replace the synthetic fertilizers with the ones that are recycled. We want to better utilize the soil microbiota function to supply the plants with the nutrients. So that means we let soil microbes make the nutrients plant available when needed. And so all this together is part of Nutri-Storm. So what we are currently doing is to look into sites where they already investigate improved soil carbon stocks. And our part will be then to look at the beneficial nutrient use efficiency and if we have an increase in soil health as a result of that. So we have a number of field trials that are not ours. So these are field trials that are from existing projects. So the one at the top here is a grassland trial in Ireland from Chagas, where we have tested or currently testing different recycled-derived fertilizers. So this comes from two projects, Nutri2Cycle and Renew2Farm. And one of these projects is using dairy sludge. And so we use dairy sludge here as a carbon-rich amendment. And we are going to test this against bitter fertilization. On the bottom, we have trials from BioAtlantis, which is an Irish company that produces various products, but one of them is also biofertilizers. So we look at the effect here on potato and onion. So other trials, again, another Chagas trial here, this time it's crop. So it's actually barley. And what we are testing here is the effect of straw return together with cover crops and slurry. We have a trial in the UK from the University of Warwick, where they look at insect frass as a replacement of mineral fertilizer. They also have a trial with barley spent grain as well, and then hopefully that picture comes up. We also have a site in Germany where they currently grow maize on rotation, where they have put in wheat straw return. They are also operating now with drought scenarios. We don't know if that is going to be relevant, but here again, we look at improved carbon stocks to see how that affects nutrient cycling, nutrient use efficiencies. All right, so one of the fertilizers, you have heard that already early on if you have been in one of the Berlin one. So insect frass as a fertilizer. So what's the idea here? So we use basically organic waste products. We feed it to the black soldier fly. The soldier fly will then be harvested in two forms. We have the larvae that turn into proteins, into animal feed. So we have that already covered. The other part here is then that they produce insect manure that can be used also as a carbon-rich fertilizer. So that is the experiment that is being carried out in Warwick at the moment, which we have already then went to the field and taken the samples for our microbial analysis. So why insect frass, apart from that the insect industry is going to grow, there have been proposed certain benefits of insect frass. So we're particularly here interested in the nutrients, but we can also assume that there may be some beneficial microorganisms, so biofertilizer potential, also some bioactive compounds, biomolecules that could not only help with growth, but also other aspects of tolerance and resistance towards pests. So this is not the focus, so we really look at the nutrient cycling, but there might be other potential benefits when it comes to insect frass as a fertilizer. Another example of our carbon-rich fertilizer is dairy processing sludge as a fertilizer. So the island is producing a lot of dairy. We have a lot of cattle. We actually have on each person in Ireland, there are 2 cattle on the island, and so there's a lot of that need to actually process these byproducts. And so we have done that in the past. So we start here with our raw dairy processing sludge, and in our case, that goes directly onto the field as a carbon-rich fertilizer, and that is the one that we are currently analyzing. But this is not the only source. We have a few other ones as well. We have already tested struvite, so that is part of our Renew to Farm project, where they also put out grassland struvites. We're currently testing hydrochar as part of that as well, and together with potato bio, that's another project. We also have tested biochar, and we have tested ash, which again goes into Renew to Farm. The hydrochar has been made by reflow, and so the dairy processing sludge that we are currently testing is coming from a project called Nutricycle. So we really kind of latching on to various existing projects that have these field trials that look at economic aspects, and we look at the nutrient cycling microbiome aspects. Okay, so what do we do with the field trial? We determine the C&P ratios. We look at soil organic carbon, total available nutrients, microbial biomass. We look at the community structure of the microbes in these soils, at the microbial capacity to turn over soil nutrients, and then we attempt to do a data integration to see which of these scenarios are most beneficial, and that will inform then our greenhouse-based targets and manipulation experiments that happen then in the second. So this is the overview of the whole project. So we are currently here in the first phase, where we have done the harvesting of the field trials. So the next stage is to actually analyze the field trials, and based on the analysis, then we gain informed scenarios of our glasshouse trials, where we do detect specific manipulations of the soil carbon stocks to see which one is actually beneficial or most beneficial for nutrient cycling and nutrient use efficiency. And then at the limit level, as we can do that in the year 30 year, we go then at field level or plot level, where we create microplots, where we then try to apply that in the field as well. So that's basically the strategy of our project. So what are the potential benefits or stakeholders? Let's move this a little bit out of the way. So there are a number of things that will be affected by, hopefully, our results. So here we have our hopefully improved ecosystem services. So we have better plant growth, improved nitrogen phosphorus uptake. We have here our improved carbon stocks. Hopefully that means we have cleaner water because we have less pollution that goes into the ground. We use fewer agrochemicals in circulation, so we have a benefit in terms of health. we have hopefully a positive effect on society, just because there's a potential to also be more efficient in food production if we think about increasing prices of mineral fertilizers. That is also then benefit for the economy because we reduce dependency on the massive import of mineral fertilizers or synthetic fertilizers. Then we have our potential improvements towards the environment, climate, so as well as then also supporting European policy, UK, EU policies for that as well. So that's almost it. Should be one more slide.
Try to click with the mouse.
Maybe it's out here. Okay. So that's basically it in terms of the project. So again, I want to highlight our partners. So one of our partners is Chuggles in Ireland. I'm from the University of Limerick. We have the University of Warwick. We have the Technological University of Munich. And we have one SME, which is BioAtlantis, which is also located in Ireland. So if you have any questions later on, you can reach me here at the University of Limerick. We also have a website now, nutristorm.eu, which is just a shell at the moment, but hopefully we'll get updated very soon. And we also have a Twitter account, so I did name it Nutristormer. Nutristorm was already used by a weird blender, so I have to put Nutristormer in it, so you can find us there as well. So yeah, if you have any questions or if you think we are missing something in terms of carbon amendments, types of analysis or stakeholders, then please give me a shot.
Thank you very much. Okay. Yes.
So I wanted to show I got a part of the presentation right. So you talked about how the project would try to develop new bio-based fertilizers using different methods, but then also talked about how you've worked on some fertilizers in the past.
So we're not developing new fertilizers as such. We use the existing ones that are used in various field trials. But what we want to do is we want to see which of these approaches are beneficial to nutrient cycling. And by reducing, so increasing nutrient use efficiency, reducing the losses of nutrients either from leaching or from atmospheric losses. And we want to make some targeted manipulations of that. So, and then at the end, we want to apply that more at the field scale, because we are aware of that. If we do that in the greenhouse, what happens in the pot trial may be quite promising, but if it's then been applied to the soil and the ground, it may not be so efficient anymore. So that is why we want to try that in the third one. So we take elements of existing trials and try to implement it.
So you are still in the developmental stage, not commercially available?
No.
Good. Thank you.
Okay, are there questions online? Peter, can you have a look?
Chat is silent, and so far no raised hands.
Okay, I think that we can continue. We go to the next presentation. It's That's Isabella from the. Okay. On. And I'm saying this now. Cecile, can you present from your laptop in the next presentation? I guess you can, because I don't have your presentation here. Probably. Okay, but we kick off now. We continue now with.
I think Cecile and me will manage this by email.
Okay, good.
So hello everyone, I'm Isabella Pinheiro. I'm from the Alfred Wegener Institute, and I'm representing the ProArmas, so producing valuable proteins and organic fertilizer from saline water sources using a recirculating multi-trophic aquaponic system. So just as a background, seafood currently supplies approximately 17% of animal protein and about 7% of total protein. And aquaculture, that means the production of aquatic organisms, will supply 6% of global seafood by 2030. And in 2022, for the first time, the farming of aquatic animals passed the capture of fisheries. That demonstrates the importance of the aquaculture sector in the world. And the shrimp, Neos vanameis, the white-legged shrimp from the Pacific Ocean, is the most economically important species in worldwide aquaculture, with almost 7 million tons produced in 2022. That's also from aquaculture and fisheries. And in Europe, the production of these species in recirculating aquaculture system is a growing industry, but only around 30% of the shrimp that we consume here comes from local production and mostly from fisheries, and the other 70% dependent on imports. And 60% of the total is from farms. So I think there's not a lot of people here from the aquaculture sector. So I brought these to illustrate the recirculating aquaculture system. So basically we have the tanks for the fish or shrimp production, and the water from this tank goes to the mechanical filter to remove the solids, the feces and the feed leftovers, and that's a part of the solid waste. So after removing these solids, the water goes to the biofilter, and then the ammonia that is excreted by the animals is transformed into nitrite and then nitrate. And then after this process goes to the degassing of CO2 and also to the UV filter for sterilization, and this clean water goes back to the fish tanks or shrimp tanks. But what about the waste? What can we do with this waste that's produced? So in aquaculture, the waste is, as I mentioned, is separated and dissolved in the water. So with a lot of nitrogen and phosphorus compounds, and also the solid waste. And through the mineralization of this organic matter, we can have also a dissolved accumulation of dissolved nutrients in this water. So the water is treated into the RAS, but there's this nitrate and phosphorus accumulation that we need to release somehow at some point. So one of the solutions is the development of recirculating multitrophic aquaponics systems, or ARMAS, that comes our name, the name of our project. And so aquaponics is the mix of aquaculture and hydroponics. So you can use the water from the animal production to irrigate the vegetable production. And in the aquaponics, there's a symbiotic relationship between farmed fish or shrimp, bacteria, and plants. So as I mentioned, the ammonia is transformed into nitrate, and this nitrogen and phosphorus is absorbed by the plants. And as advantages of this system, we can have an improved quality and reduce water use, and also improve profitability because we have an economic diversification with the production of different crops. But to use this concept in marine effluents, so we need the use of halophyte plants. Halophytes are salt-tolerant plants, so they can grow in a variety of saline environments, such as mangrove, salt marshes, and they can tolerate up to 60 ppt of salt. And these marine aquaponics, it was developed as an alternative for freshwater aquaponics due to the limitation of water use and also soil salinization. And so the integrated culture with halophyte plants has been studied for treatment of brackish and seawater aquaculture effluents. So the goal of ProArmas is to develop a recirculating multitrophic aquaponics system as an innovative food production system that will close the cycle, produce highest quality protein, and maximize water and nutrient use efficiency. So to achieve this goal, we are going to produce shrimp as our high value product. The kilogram of shrimp here in Germany, for example, is being sold from 60 to 100 euros the kilogram. And so to treat the waste of this shrimp production, we are going to use the halophytes that we are going to produce for human consumption. We are going to produce saliconia and also other different types of halophytes, but saliconia is the one with the highest value in the market, costing around 30 euros the kilogram. And then we are also going to produce polychaetes because they have a high protein content, almost 50 to 60% of protein. And then we are going to use this biomass as an alternative to fish meal in the shrimp feed. And also the macroalgae we are going to use as a feed additive due to the contents of vitamins and minerals and antioxidant compounds. So these two biomasses we are going to add to produce an innovative feed for the shrimp. And then at the end of our project, we are going to produce the demo farm. And I'm going to talk more. So the ProArmas, the goal of ProArmas is meeting two of the topics of the Green Arrow Hub call. The first one is enhance fertilizer efficiency and reduce fertilizer use. And the second, increase European protein as self-sufficiency. Due to the circularity of the system, then we can have maximized nutrient reuse and also protein production. So the coordination of the project is at AVI with us, and then we have two partners, the Klaipėda Science and Technology Park in Lithuania. They are going to be running the trials with the feeds for the shrimp production. And then we have also the University of Padova in Italy, and they are going to run the studies with the halophytes, so aquaponics and also hydroponics. And we also have an industry partner that is the Green Hub company here from Germany, and they are going to build the system for the polychaetes production. So the objectives of ProArmas is produce innovative protein source in armas, optimize the polychaete and macroalgae production, develop sustainable feeds for shrimp and Eusbanamei using polychaetes and uva, evaluate the use of live polychaetes and fresh seaweed as dietary supplements in shrimp feeding. We are also going to run these experiments. And produce alternative biofertilizer for the shrimp production that will be tested in the growth of novel vegetable species. Design and implement a demo farm for the integrated production of shrimp, polychaetes, seaweeds, and halophytes. And evaluate the environmental and economic sustainability of the armas. So to achieve these goals, we have 4 work packages, technical work packages. The first one is the biggest one, is integrating and producing innovative protein sources that we are going to run at AVI. This basically is conceptualize, design, and construct the systems for polychaetes and algae production. And we are going to test different substrates, different ratios of waste that we can use both for the polychaetes and for the plants, and also we are going to test some light regimes. So then we are going to use this biomass to produce our feed and also to send to Lithuania, where KSTP will run the experiments with the shrimp for alternative protein sources in aquafeed for the valorization of sustainably produced polychaete and uva. And then we are going to send both AVI and, because we also have some shrimp production here, and the partners from Lithuania, we are going to send the waste to Italy so they can test the biofertilizers for the plants. And then at the end of the project, we are going to create the demo farm for resilient, sustainable, and valuable food production. We at AVI, we are coordinating this work package, but it's part of the work of everybody, of course. And we are going to try to combine the different requirements of the organism that we are going to produce together to create a sustainable farm. So the main stakeholders of the project is industry and enterprises, horticulture and aquaculture sectors, biotechnology, feed manufacturers, scientists, and companies interested in human and animal nutrition and closed loop waste recirculation. And regarding the timeline, so we just started the project. We are just trying to combine the system for the polychaetes. We probably are going to start to produce the polychaetes soon, next month is our plan. And then we are going to, next year, we are going to deliver the biomass of polychaetes and to KSTP so they can start the trials with the shrimp. And then in 2026, it's the delivery of shrimp sludge to Italy. Of course, everyone is doing the trials, the previous trials during these 2 years, and no one is stopping. And at the end of the project in 2027, we need to present the demo farm and also organize the workshops and training at AVI. So the expected impacts, the project will impact consumers as it will allow the development of a short chain production system able to offer sustainable food produced close to urban centers. And the main categories benefiting from ProArmas will be, of course, university students, school pupils hosted during the guided tours that we are going to have in our demo farm, technicians, farmers and entrepreneurs, civil society through open science events, and policymakers engaged in the final conference and involved in outreach activities. So I just wanted to show, we already had lunch, but to show you the use of the salicornia, the halophyte, and this is came from an integrated production of shrimp and salicornia that I carried in my doctorate in Brazil, and the beer was actually produced with biomass that I produced it there, and it was really good. So thank you.
Yes.
How much can you control the system? I think in terms of temperature, especially, I think there's
yes, this is something that we need to think about it because, of course, they have different requirements. The shrimp needs almost 30 degrees of water, and the Ulva strain that we are going to use is from the North Sea, so around 10 to 15 degrees. Maybe we'll have to come up with a decoupled system, so they are integrated, but not in recirculate.
Hi, Manuel from Pasha. I'm a Luxembourgish aquaponic company. I saw a little bit of maybe of a challenge. I think, according to my knowledge, EU regulation requires 3 degrees of difference between one animal and its feed. But right now you're assuming that the waste of the shrimp can be used to feed the animal that will go back to feed the shrimp. So there's only 1 degree of difference. Do you think that's going to be an issue in the future?
I will have to check this info. I'm not sure about it.
We'll talk.
But good to know.
Have you think about the risk of contamination with human pathogens, for example, bacteria like Escherichia coli or something like this in this circulation system?
Normally, we don't have this problem, but it's of course something that we have.
The temperature is much high, so
normally in the shrimp production we don't have this problem.
no.
But it's something that we will have to analyze throughout the project.
Wonderful. I think we continue. Thank you very much, Isabel. Okay. Petra
Yes.
and Cecilia.
Cecile
Yes.
is there and she should
Yes.
be able to share the presentation.
Okay.
And meanwhile, a message.
I think from now onwards we have everybody online will share, right? No, I think oh no, it's not true. I have one more here. Okay, Cecilia.
Yes. Can you see my slide?
We can
Yes.
see the slide, but not yet in presentation mode.
No?
Yeah, perfect.
Okay. Thank you very much.
Okay, how can I make this big here?
You have to say share whole screen.
Yeah.
That's what I did.
I'm talking to
Okay.
Christian,
Oh,
sorry.
sorry.
Okay. Good question.
In layout, at the top, there's this layout button.
Yeah.
Top.
This top.
Ah, here. Oh, okay.
Maybe
Maybe that one. Hmm.
Yeah, you can close this one maybe.
Yeah. And then there's these two arrows on the right. You see
Oh, no, do this one.
The corner.
in the corner, right?
This one. Yeah.
Yes.
Good. So that the next project is Vertigo, and then
Yes.
Cecile from
Belgium.
also the Free University in Brussels.
Yes.
Good.
So thank you, and again, good afternoon. I will present this Vertigo project, which title is Improving Fertilizing Strategies by Innovative Use of Plantain in Agriculture Systems. I hope it works when I... Sorry. So the background of this project is, as explained in the pitch, is the non-sustainable system of certain agro ecosystem where nitrogen and phosphorus inputs are sometimes not efficiently used by the crop of interest. This is mainly used due to the nitrogen cycle, where we have, in some cases, intense nitrification rates and denitrification rates that can form nitrogen forms that can easily leave the system, such as nitrate that can leach out from the soil, and nitrous oxide that can be emitted as nitrogen gas, greenhouse gas. Phosphorus, again, as I explained in the pitch, we have phosphorus that when it is provided to the system can be easily immobilized in the soil matrix and therefore not available for the plant of interest. In our project, we want to improve this system and to have a better use efficiency of both nitrogen and phosphorus. How we want to do that is by using service plants for improving nitrogen and phosphorus use efficiency for cash crop. So on the left side, the trait that we are going to use for the service plants is through this biological nitrification inhibition ability of certain plants, such as plantain, that can produce certain compounds either exuded from the root system or accumulated in the plant tissue. And these compounds have been shown to slow down the transformation of ammonium into nitrate. On the other side, we have other plant species that can also, through their rhizospheric function, mobilize phosphorus that are either bound to the organic pool or to the mineral pool. So again, this slide showing you the nitrogen cycle. You see the different step in the transformation of nitrogen. And here you see that plantain as a service plant can emit these biological nitrification inhibitors that will inhibit the key processes, reducing therefore the nitrogen losses from the system. For the phosphorus, we also know that certain plant species can, through their rhizospheric function, mobilize phosphorus either by producing, for instance, phosphatase that will degrade organic phosphate forms, or through, for instance, low molecular weight organic acid that will really solubilize phosphorus from the mineral pool. As I said in the pitch, we have different targeted agrosystems that will be studied by the different partners of the Vertigo project. Temporary grasslands, so this is really where we want to see if the plant composition of this grassland when integrating planting can really help slowing down the nitrification rate. We also study planting as a part or as a mixture or part of the mixture of cover crop that is afterwards followed by spring cereals. And we also have plantains and other phosphorus mobilizing plants that can be associated to maize as companion plant. Scientific work packages, we have 4 of them. The first one aims to develop Plantigo-based companion crop mixture as a to improve both nitrogen and phosphorus nutrition of the target spring cereals, which is here in this case maize. So as I said, we want to identify species that can, on one side, slow down the nitrification rates of nitrogen, and on the other side, that can improve mobilization of phosphorus. The idea is also to improve the provision of phosphorus and nitrogen that were provided from after the integration of catch crop. The second work package, which is actually our third work package, there we want to incorporate plantain, not necessarily in classical cereal-based rotation, but rather in integrated arable and livestock systems. So on one side, we will target temporary grasslands, and on the other side, so crop mixture, there was less livestock integration, but also a bit like in the Catch BNI project, we want to see how plantain, when it is in the plant composition of the preceding crop, in the cover crop, can really benefit for the following crop. In this case, it will also be barley for the Irish colleagues. And the last scientific work package is aiming at optimizing organic fertilizer, also with a Plantago-based approach, where the idea is, like also in the Catch BNI project, we want to see how organic fertilizers such as digestate, slurry, manure, animal manure can be stabilized if we add plant material to it to stabilize and to reduce the transformation of, on one side, the organic form of nitrogen and the ammonium conversion into nitrate. So this project just started in June, actually, so we don't have any results yet at this stage, but some impressions. So for the first scientific work package, we started with the screening of more than 60 plantain genotypes, using an hydroponic and a soil-based approach for doing the screening, using a pipeline that we developed actually in the frame of the Catch BNI project. So now we are applying this pipeline in particular for plantain. And then on the other side, on the right, you can see by the colleagues of IPK in Germany, hydroponic screening for phosphorus mobilizing properties of different plant species. So in particular, Phacelia, raygrass, French and German raygrass, and red fescue. We also have a strong stakeholder engagement strategy and a communication strategy, and for that, every country participating in the project is creating a national stakeholders board. So this is almost achieved, and the idea is to really have a co-creation discussion with every of the stakeholder boards, so that we can design our experiment so that it fits the reality. At the same time, we are developing questionnaires for farmers and advisors, again, in a co-creation way of thinking. And for that, we are going to use the Lime survey tool. So this is also in process and soon, released. I cannot. Hello?
Can you just try to reshare?
Yeah, I don't know why. Someone. Yeah. Can you see my screen again?
Yeah.
Yeah. And last but not least, we also developed communication tools. We will have soon released the Vertigo, the poster, some Vertigo flyers, and we also are going to have 2 times per year, or at least once per year, a Vertigo newsletter. Oops. Yeah. Thank you for your attention. And here you can see the names of the different PIs of this, from the different partners of this project. Thank you.
Thank you very much, Cécile. Good.
Questions?
Yes, any questions? Any questions in the room?
Yeah. Why did you choose hydroponic as a screening for the genotypes? If you have a mixture system later with maize, I think it's a total different routing and soil condition.
So the question is why we are using hydroponic as a tool for screening?
Yes, yeah.
Yeah, because when we are using a soil-based system, you are pretty limited to the nitrifier community that you have in that soil. So in that sense, if you use one day one soil and another day another soil, you could probably have different nitrifiers and denitrifier community. So to get rid of that and to be really in a control condition, we also have an hydroponic system where we developed this pipeline so that we are sure we are collecting always in the same way the exudates from these plants, and then we process these exudates and we submit these exudates to a small bioassay with a model nitrifier bacteria, and we see how much nitrate is produced when we provide ammonium to this bacteria. And the less nitrate, the more inhibition you have from the nitrification. So this is an approach that is commonly used in the BNI community. And together with that, we also have soil-based where we monitor the change of the different of the nitrification by measuring also on one side nitrate in the soil and also N2O emitted from the soil. But indeed, just having a hydroponic approach would be very limiting. So that's why we also developed a soil-based approach to do this screening.
Okay, I think you answered the question, but there's one more question.
Yes.
Hi, Cecile. Silvia Matassa from University of Naples and in Italy. I was wondering, can you actually also measure the amount of exudates that these plants are producing in your system and therefore estimate how much of these exudates are needed to actually inhibit the nitrification activity, or are you planning to do so?
I don't know how you could really, actually the exudates are, it's hard to say how you could really quantify. So maybe you could quantify the carbon content. This is what you mean?
Yeah. Some indicators that would say how much exudates are actually produced and under certain conditions.
Yeah, we also have in this project collaboration with another project where we will characterize the exudates by chromatography and eventually even characterize the molecules that are responsible for the inhibition of nitrification. So this is also planned. And for the second part of your question, for this applied project, it's not yet planned to have such optimization. So that will maybe come later. But that's a good suggestion.
Okay, thank you and good luck.
Thank you.
Okay, I think we're very good with the time. We have one more presentation to go, and I have the presentation here. Fiona, you with us?
Yeah, hi.
Very good. But I need to think, I think I need to share my screen here.
Yeah, or I can share whatever is easy.
It's okay, I'm already.
Fiona should be able to share.
It's okay, I'm sharing it already.
Okay, great.
Okay. Okay, we're leaving the plant system a little bit and we're going to the animals. Fiona, all yours.
Thank you. So my name is Fiona, and for the next few minutes, I will be talking to you about our Sustained Sheep project. So within this project, we are looking to reduce methane emissions in sheep via genetics. If you want to move on to the next slide. I'd like to acknowledge the project partners. So as you can see, we have partners from Ireland, the United Kingdom, New Zealand, France, Norway, and Uruguay. So it's quite a diverse group working within the project, but it should make for some interesting data collection. You can move to the next slide. Over the next few minutes, I will give you an introduction to the project and to why we have decided to carry out this work. We will look at our objectives, how we are going to measure the methane from the sheep populations across the various countries, the impacts we expect from the project, and our stakeholder inclusion model. Next slide, please. So as I alluded to in my pitch earlier, sheep, albeit small, are a contributor to global greenhouse gas emissions, and they're responsible for about 7% of global emissions annually. So obviously a lot smaller than dairy cows or beef cattle, but still significant nonetheless. Previous research carried out by partners within our project has shown that there is genetic potential to breed animals for improved productivity, but also for reduced methane output. And I guess that is quite important because in each of the partner countries involved in this project, there are quite stable genetic basis for breeding programs within their sheep populations. So farmers are selecting animals and specifically sires based on their genetic potential to breed the next generation. So if they can do that, but also select an animal that's potentially going to produce less methane, it's favourable throughout. Genetic improvement is probably an underused mitigation tool across countries just because of, I guess, the value in selecting the animals and then also like what it's worth back to the industry. So we want to put a big effort in our project on discussing this with policymakers within each of the countries and by doing a lot of communicative work with our stakeholders in order to, hopefully increase the use of genetics as a mitigation tool. Next slide, please. So there are three main objectives for the duration of the project. We want to integrate low environmental impact traits such as methane output into national breeding schemes in each of the partner countries. Our aim is to use portable accumulation chambers, which I will refer to as PACs, to measure methane from sheep. And then we want to look across countries at standardizing genetic improvement, both as a mitigation tool, but also in the development of genetic gain for sheep populations. Next slide, please. So I mentioned earlier that methane has been shown to have a genetic potential and be influenced by the genetics of the animal. We looked at the heritability of the trait. So that basically means the proportion of the trait that's down to the genetics of the animal. And when we looked at it, we looked at different variations of methane as a trait. So the first one there, when you see grams of methane per day, that's like absolute methane output. But then we also looked at the methane output of the animal as a proportion of their feed intake, the amount of CO2 that the animal is emitting, and then we looked at combinations of methane and CO2, which in some countries they are using as predictors of feed intake in ruminant animals. And then we looked at methane output as a proportion of the animal's live weight, so a more efficiency trait. What we saw, if you just look at the averages, across the studies, all of the traits that we looked at were heritable, so there was genetic influence in each of the traits. It was higher for some compared to others, but absolute methane did look favourable. And at the moment, it's probably up for discussion, but we're tying between the idea of either including absolute methane as a trait for itself or perhaps some form of efficiency trait, so methane as a proportion of dry matter intake or as a proportion of animal live weight. Next slide, please. So I'm not sure if this is going to work. Oh, there we go. The video is playing. So this is just showing how our pack chambers work, so how the animals are measured for methane output. So it's a trailer essentially, and there are 12 boxes on the trailer, and then the animals, so one animal enters each box for a 50-minute period. Within that period, we measure methane output, CO2 output, and oxygen consumption, and the measurements are taken at 3 time points while the animals are in the box. So what it allows us to do is it allows us to rank the animals based on their methane output. So you're not getting like a daily absolute methane output as you would with a respiration chamber or perhaps a green feed device. But you are getting a ranking of output across animals within a population, which for genetic purposes is an ideal measurement. Next slide, please. So we're quite lucky that each of the countries involved in this project have packed chambers, so the measurement technique throughout will be the same. What impacts or outputs, I suppose, are we expecting from the project? We're hoping that it'll increase efficiency across sheep farming systems. If farmers are choosing animals based on their genetic potential, then these animals will be more efficient, they'll have higher levels of production and lower levels of methane output. We want to generate tools for farmers and for industry stakeholders to compare methane emissions either between animals within their own flock, but then also for animals across different flocks within a similar population. And our aim is to, I suppose, promote genetics as a mitigation tool within ruminant animal species, but then also to support policy measures and to increase awareness and adoption of genetics as a mitigation tool. Next slide, please. So from a stakeholder's point of view, it is quite important to us, and it's probably a big aspect of the project, that we communicate with our stakeholders continuously and to bring them along with us. The graph might be a little bit confusing, but we have, I suppose, stakeholders across a range of groups, I guess. So like all the way from farmers, animal breeders, right through then to like scientists and policymakers, and obviously to customers as well, because the farmers that will be using our tools are essentially producing products for human consumption. So how do we plan on doing this? We'll have regular stakeholder meetings and update groups for our stakeholders. We're producing a newsletter. Our project just started in late spring of this year, but we've produced our first newsletter already. We have a podcast for the project, so that's like open for anybody to listen to, and then we'll obviously have scientific outputs as well.
Okay, thank you.
No problem. I'm
Are
not
you
sure.
still there?
Yeah, I'm.
Okay, okay, couldn't hear you.
So this slide didn't change on my screen, but I'm not sure if that's just my computer, because I can see that it has changed on yours. But anyway, just want to say thank you to everybody for your contribution, and if there are any questions.
Okay, are there any questions?
Yeah, maybe. I know from dairy systems that they have the mobile chambers or they measure the methane when they are milk in the milking time to reduce stress. How do you deal with this stress factor if they are put in these small chambers? They may be stressed, and I know if you have an animal experiment, you have to mind about the welfare and those aspects. How do you deal with this aspect in the project?
Yeah, so from a welfare perspective, we have both an in-house, so within Chegas, we have a welfare committee, and then nationally we have welfare guidelines as well. So all of the techniques that we're using are approved by both of those bodies and licensed for animal use. We have been using the chambers now since 2019, so over the last 5 years, and we have never had an issue with animal stress. Once they go into the box, there's a clear, like a window on the front, so they can see the animal next door to them, and they can obviously hear and smell the other animals as well. And they actually seem quite relaxed inside. Like you'll often see them lying down, just chewing their cud while they're in the box. I know in New Zealand, one of our partner countries, their ethical committee actually got people to sit in the boxes for the same length of time as the sheep, just to kind of have a grounding of what it felt like, and that was part of their like ethical process.
Are there any more questions from Paris? No. I have a question. So, how do you, let's say, interact with the, let's say, cattle community? You know, there must be people doing similar work with cows, or is that
Yeah,
not the case?
yes, yeah, there definitely is. So I know the organization that I work for, Chagis, we have dairy, beef, and sheep departments all working together. So we would work alongside the scientists doing similar work with dairy and beef. We have in Ireland already implemented methane within our genetic indexes for dairy and beef. So now, from a sheep perspective, I guess we're just following on.
Okay. We still have 1 more minute. So I have another question. And but that's now completely different. So you're trying to reduce methane emissions from the sheep by, let's say, looking at the whatever genetics of the sheep. When I used to do my PhD, and it's 20 years ago, I remember that there was in North America, there was a Nobel Foundation or something, and they looked at basically at the plant side, you know, which was fed to the cows. So the feed for the cows, they tried to, you know, modify that so that they would produce less methane. I don't know, did that completely fail?
Yeah,
These
no,
kind
not
of
at all. We
Okay.
have other work ongoing on different projects, also looking at dietary components. So
Okay.
in Ireland, we're like a mainly grass-based country, and we have seen that if you include clovers like white clover, red clover, or herbs like chicory into your sward, the animals are actually ranking lower for methane output than if they were just eating a plain perennial ryegrass-based sward.
Okay. Good. So I don't know if you've all seen this. We will continue. We have a coffee break now, or tea break, whatever you prefer. And we will start the last pitch talks at 4:10. And so we have 35 minutes, very generous. Okay, so thank you for the people online, and thank you for the people here, and especially thank you for the presenters. We see you soon.